Why don't we just keep going here? So starting with the same thank you slide, and really got to say, the team at Cognition is just absolutely stupendous. These are the people who did all of this work, and they're all here. They all deserve a round of applause because I just send emails for a living, so, that's how it goes. I just wanted to let you know we've had the great fortune of receiving quite a lot of support from NIA, both for the preclinical studies and the clinical studies. So this is really important to acknowledge the significant role that NIH has played in the absence, or I should say in the presence of quite a lot of clinical trial failures in this sector. Without them, there really wouldn't be much going on. So I just wanted to remind folks the rationale for targeting A-beta, because this sometimes gets lost and the baby gets thrown out with the bathwater in a lot of the discussion and debate that happens these days. So there really is overwhelming genetic evidence of A-beta's role in Alzheimer's pathology, not only from the early onset mutations where more than 200 of them really actually impact the concentration or the assembly rate or the ratio of A-beta protein, but also from the late onset risk genes, the biggest one of which is ApoE, that is the main clearance pathway for AD. We think that the reason why previous therapeutics have really failed is that A-beta can exist in a variety of structurally distinct forms, and Aladdin touched on this, but I thought it would be good to go into a little bit more detail here. What you're seeing is the structural equilibrium that actually has a Y shape in between fibrils on the one hand and monomer, which is an intrinsically disordered proteins, and oligomers, which are literally technically a metastable intermediate state. And so when you look at the therapeutic history of what folks have done in Alzheimer's disease, they've sort of tried to turn off the tap by inhibiting the two enzymes that cut A-beta protein out of the precursor. And so this would be gamma or beta secretase inhibitors. And so they do, in fact, effectively inhibit monomer production. This has been shown in very many studies, so they're very effective at what they do. But they had no effect on disease progression. And in fact, folks got cognitively worse, which was really a shame. For many of these inhibitors, the fibrils certainly remain in the brain as a source of monomer. So the fibrils can dissociate back to monomer and then go on to form more oligomers, especially at locally high concentrations you see around synapses. So on the other side of things, folks have tried monoclonal antibodies. And so these are antibodies that are directed against A-beta. And they did, especially in the case of aducanumab, a really effective job at clearing fibrils and plaques out of the brain. Some of those folks had fibril and plaque load that was reduced below the level of detection with amyloid PET agents. So it was really quite remarkable, but there was no effect on disease progression. And we think it's because those antibodies don't really engage with the oligomer target. So they're really not effectively dealing with the toxin that's causing disease. So Nick described to you our drug. It sort of sits in the middle of this equilibrium, and we've demonstrated in a variety of ways that 1812 can actually selectively displace that oligomer out of that binding pocket without having any effect on the monomer concentration. And that's key. So you really don't want to touch that monomer, and you really don't want to perturb what's going on in its normal physiological role, even though it's not very well characterized. We know that it's secreted in an activity-dependent manner at the synapse, and that usually means that it's doing something pretty important. So we and other folks have demonstrated that that oligomeric form binds to a single saturable site at synapses, that this causes the toxicity at picomolar concentrations in Alzheimer's brain. And what we identified through this phenotypic approach was an intervention point. So the point where you can stop the downstream toxicity that flows from it. And I won't go into this, nor will I spend a lot of time on this slide either, since Nick did a very effective job at guiding you through the preclinical evidence that suggests that all of that's true. So we went from our preclinical studies to first-in-man studies. This was a pretty routine single ascending, multiple ascending dose study in healthy young and elderly volunteers. And we saw that we had safety and tolerability at doses that were expected to produce therapeutic concentrations. This had been defined from the preclinical animal model studies as something that exceeded 80% receptor occupancy at the receptor in the brain. And so we then went on from there to our first in-patient study. And Nick showed you some of the data from that. This was a very short study in which we saw a lot of changes, including a reduction in synaptic damage markers. And you've seen some of this previously, so I'll go through this fast. Again, this is the study design, very short, 28 days. So this was the first patient exposure. And in 19 patients that were randomized to one of 3 doses of drug or placebo that was given by mouth once a day for 28 days. So I'm going to be showing you data from the spinal taps, so the protein expression in the spinal taps that were done at baseline and at day 28 of the study. So the first thing that we did was look for oligomer concentrations in the CSF. And so you remember Nick had taken you through that preclinical with the red dots, the graph all the way over on the right side. After a single dose of drug, we see an increase in oligomer concentration that's dose-dependent out in the CSF. And so this was pretty unexpected that we would see the drug not only displace it from the receptor site, but somehow facilitate clearance out of the brain, either directly or indirectly. We still don't know which is the case. So we went ahead and looked into patients, and the results from the trial are shown in the middle. On the y-axis, you're looking at the change from the patient's own baseline, between baseline and day 28. And the placebo in the diamonds and the hex is the pooled treated patients. And so what we saw was a significant increase in the concentration of the oligomers in the CSF compared to the patients on baseline. We didn't see that in the placebo. The difference between the 2 groups was significant. And so this was consistent with the drug's mechanism of action, consistent with what we had seen in preclinical studies, and we thought encouraging evidence of target engagement. The drug had gotten into the brain and engaged the target. I've also spoken to you in the past about the protein expression changes that we saw out in the CSF as well as the plasma. Quite a number of proteins that are changing in pathways that are known to be impacted by Alzheimer's disease. We also took a look for specific proteins, targeting those proteins that are markers of synaptic degeneration. So this is an effort in Alzheimer's disease to really understand different classes of markers and what it's telling you about the degeneration that's going on in the brain. And so in the box on the left-hand side is a figure from the literature. Quite a number of folks have looked at CSF expression of fragments of synaptic proteins. In this case, it's neurogranin. This is a postsynaptic protein that's critical for synaptic plasticity. You can see it in the neurons in the dish in our preclinical studies on the right-hand side. It's normally really robustly expressed not only in the dendrites but also out in the spines. When you add oligomers, it just wipes out expression. But when you get rid of those oligomers by displacing them with the compound, the expression just is restored back to normal. So we knew that we wanted to take a look at this, and we did, and the results of the trial are shown in the middle. This was an analysis that was performed by Kai Blenoff at the University of Gothenburg. And so what you can see, same kind of graph in the middle there, change from the patient's own baseline in the ELISAs that Kai did with the change in the neurogranin fragment concentration increasing in the placebo patients and decreasing in the compound-treated patients. So this was not the only synaptic protein that had this pattern. We also saw something similar for synaptotagmin. Again, the same kind of pattern. In the literature, we know that fragments of synaptotagmin go up as the disease progresses. We saw restoration of synaptotagmin expression in brain cells once you get rid of the oligomers. The results of the trial are in the middle, the same pattern. The drug-treated patients have a decrease in their CSF concentrations of synaptotagmin fragments compared to their own baseline, whereas the placebo patients increases, and the difference is significant. So lastly, we wanted to look at tau, and so this is new data. since the last time we spoke to you. Tau is obviously hyperphosphorylated tau is the second major histopathological correlate of Alzheimer's disease. The neurofibrillary tangles are composed of tau molecules that are absolutely loaded at dozens and dozens of sites with tau. And so we know from the preclinical literature that the story is interesting, and it's starting to get more interesting as time goes on. So we know that when you put A-beta oligomers on neurons in the dish, you see an upregulation of tau kinases, and you start to see phosphate loaded onto the tau, and tau mislocalizing out of the axon into the dendrite, where it participates directly in synaptotoxicity. So we went to a colleague, Ian Pike at Proteome Sciences, who has a tandem mass spec method that allows him to look at where the phosphate is along the 400 plus amino acid length of tau. So folks are starting to look at this. Where is the phosphate in that sequence? How many of them are phosphorylated? Because we think that the tau biology is going to be considerably more complex than simply what you would see at the end stage with the neurofibrillary tangles. So Ian has a method to look using tandem mass spec at both unphosphorylated and phosphorylated protein and reliably quantify 35, 36 of the known 39 sites where phosphate can sit on tau. And what they published, this is a study that they published in 2017, and here what you're looking at is a ratio between the Alzheimer's patients and the age-matched control patients, and the height of the bar indicates the degree of phosphorylation. And as you would expect, you see an increase at 11 out of those 35 phosphorylation sites. That's increasing by at least 30% above the noise level, which is the noise level, in the 80 patients compared to controls. Okay, great. This is as expected. This tells you where it is along the length of the tau molecule. So we went to Ian, and we asked him to do the same thing in the CSF samples from the patients in our trial. And so the results are in the next slide. Again, on the left-hand side in red is that same figure I just showed you, the Russell paper. And the same lab, same method, same machine, same instrumentation. These are the CSF samples from our trial. And so here, what we're looking at is the ratio of the drug-treated compared to the placebo-treated patients. So you're looking at, as Tony likes to say, a delta-delta. So this is the change from the patient's own baseline, and now we're looking at the median for the two groups expressed as a ratio. It's a little bit complex, but it's meant to reflect and allow you to easily compare to the Alzheimer's patient graph, the published graph. So what you can see is that 6 out of those 33 phosphorylation sites decreased by at least 30% in the drug-treated patients compared to the placebo-treated patients. So that's a pretty broad reduction at a number of amino acid sites in the phosphates on the tau molecule that winds up in the CSF. So there's really only one way that that can happen, and that's if the drug is acting upstream of the kinases, those 5 or 6 kinases that are loading phosphate onto tau. So this is the first clinical evidence of a broad reduction in phosphorylation at a number of amino acid sites at tau, and it supports the A-beta oligomer hypothesis of Alzheimer's disease. It suggests that it's the A-beta and the A-beta in the oligomeric form that's setting in motion the changes that lead to what we see going on with tau. It certainly is consistent with observed reduction of synaptic damage, so that abnormally phosphorylated synaptic tau, which is something that Tara Spires-Jones' lab focuses on and studies quite extensively, which itself is directly synaptotoxic, is potentially being reduced by the compound. So I'll go a little bit more rapidly. So evidence, encouraging evidence for target engagement Nick showed you evidence for disease modification, and we think encouraging evidence that there is synaptic protection after only 4 weeks of treatment with the compound, because we see a reduction in these damaged proteins as well as tau phosphorylation itself. So we've got a number of clinical trials going on right now under the able direction of our brand new Chief Medical Officer, Tony Caggiano. Wave, Tony, so everyone can see you. I didn't introduce you before, but there he is. We're very fortunate Tony joined us. And so those trials are looking at, I'll show you the clinical trial design in a second. Those trials are looking at various aspects of the kinetics with which the oligomers move out of the brain into the CSF. And we're also looking at, in more detail, at synapse number and are conducting some really innovative clinical science with all of these, as well as a large efficacy study. So really briefly, I'll take you through a couple of them. This is the study where we're going to be measuring CSF fluid hour by hour after a single dose of drug, and we're looking for the kinetic response of those oligomers. We're looking to measure the oligomer response over time in the CSF of patients. This is a placebo-controlled trial, like all of the trials we've done, in which we're looking at either a single dose of drug or placebo to compare. So we're pretty excited about this one and what it can tell us about oligomer dynamics over time, rapid dynamics. We're also doing the world's first trial using the UCBJ imaging agent. This is a derivative of levetiracetam, and that's a drug that binds to SV2A proteins, an anti-epileptic drug. And the folks at Yale, Richard Carson and Chris Van Dyke, discovered that this is just an excellent PET tracer, excellent kinetics, and it can quantitatively image synaptic density in the brain of a living patient. So in this trial, which was originally 6 months and has now been extended for a further 6 months, what we're doing is looking at patients with mild to moderate Alzheimer's disease that have been treated with 1 of 2 doses of drug or placebo. And we're going to be doing an awful lot of imaging on these patients. We're going to look at synaptic density with UCBJ at baseline, 3 months, 6 months, and again at 12 months. We're going to be doing FDG PET at baseline and 6 months and 12 months. We're also going to be looking at resting state bold throughout the trial, as well as taking the cognitive tests throughout the trial. And we'll do the spinal taps at 0 and 6 months and hopefully get a lot of volunteers at 12 months so that we can look for the first time at changes in synaptic proteins in the CSF, changes in synaptic density centrally, and how both of those relate back to behavioral performance in the same patient over time. So we're really excited about this one. And so the efficacy study is the same 2 doses. We've got 2 cohorts here. Part A, I'm very happy to say, because of the just incredible results, incredible effort of the folks in this room, the last patient is going to get their final spinal tap from that Part A cohort of patients October 24th. So sometime in the new year, we should have results to report back to you guys about what we saw after once daily 6-month administration of Aleta for the first time. Same mild to moderate patients. We're focused on that throughout the clinical trials so far. Part B is just sort of scaling that up. Not as much imaging in this study, but we will be doing the cognitive testing and the spinal taps to look at the protein expression. And then finally, our brand new study, which has a brand new name, SEQUEL. So this is a supportive physiology study in which we're looking at the same thing we did initially, just a 1-month exposure with a washout and then a crossover. So this is a double-blind crossover study in which we're looking at the higher dose of 1812 and looking at its ability to impact theta power. So this is one of the EEG rhythms that's affected in Alzheimer's disease. So with that, I'll end hopefully early and take any questions you have.
So, very interesting presentations. Congratulations on the work to date of you and your team. I wanted to ask probably what is a very naive question, but is there any reason to believe that the compound Eleta has effects on its own, absent A beta? And the reason I say that to also suggest that, or ask whether there's an appetite to measure microvessel blood flow. Just this year, Dave Atwell from the UK published that A beta actually acts in part by constricting the microvasculature, thereby resulting in diminished energy to the brain and causing all kinds of things. So I wonder whether, one, you might look at your compound on maybe some cerebral blood flow endpoints, but two, to ask the question, is the compound working only under conditions of amyloid overload, or does it in and of itself have any positive effects?
Sure. It's a very good question, Sunny. So we don't think that it interacts with anything else or on its own. And there's a couple of reasons for thinking that. First of all, the standard counter screening that we did at, I think, 100 receptors shows that we're very well separated 100-fold versus affinity at anything else. So that's not the universe of proteins, certainly. It always is a formal possibility, but we think this is truly selective for Sigma-2. Secondly, the way that we did the phenotypic screens initially, and that is what drove SAR, was to always test whether the compound was neuroactive on its own. So it was very important to us, since we knew that synaptic plasticity mechanisms are extremely sensitive and likely to be negatively impacted rather than positively impacted by any CNS active pharmacology, that we rule that out. We were deliberately looking for something that only affected the disease state and that did not have a nootropic effect, that did not augment normal learning and memory or anything like that. So as we all come to grips with Sigma-2 pharmacology and the state dependency there or the interactions of these pharmacology sites with each other, this is really interesting, but it is something that we deliberately took off the table at the very beginning.
I just want to add a comment to that. Some of the other activities that you've seen people talking about today, such as metabolic stimulation or caspase activation, that was part of our screening process too, and we selected, we have seen compounds that have those activities, definitely, and part of our screening was to select compounds that did not have those type of activities, but did have the anti-A beta activities.
Should definitely listen to Nick because he's the one who does the work.
Excellent talk. I wondered if you had a chance, I know there's a lot of data to sift through, but if you looked at all of those specific phospho-tau sites to see if they've already been cataloged, if you will, for how it affects the tau function?
This is an absolutely fascinating and emerging aspect of what we're all starting to look at in Alzheimer's disease. So the more we know about tau, the more we know that it's an extremely complex molecule. And what we had previously done through the history of Alzheimer's disease was sort of look one ELISA at a time. So one amino acid pair at a time, one amino acid site, one phosphorylation site at a time. And that's not given us a really good picture of this dynamic molecule and its potential roles, the potential roles of the phosphate at the different amino acid sites for triggering downstream biology. So I think the profession is just at the beginning stages of that. There's some work about to be published about using these kinds of tandem mass spec studies to look at the entire length and where the phosphate is along it. Obviously biofluid samples give you one perspective on that versus preclinical studies where you could really, get the full picture of what's going on. But it's a very active, important molecule and certainly can be directly synaptotoxic under some conditions. Okay. If there are no more questions, then... sorry. I'm sorry, Carmen.
What do you expect from the FDG study, the one that you will do with FDG? Because it is not the right, maybe, marker for CNS. So I don't know what you are going to expect with this PET.
Sure. So we included FDG PET. Certainly there's a long history of looking at this in Alzheimer's trials. There's a certain power associated with the hypointensity that's seen in Alzheimer's patients. It's a very dynamic readout of quite a number of things, glucose utilization, blood flow, oxygenation, a variety of things. But it was the canonical marker of synaptic function in the brain. And so comparing that directly to SV2A, this new synaptic density marker, was one of the rationales for taking a look at that in conjunction with the SV2A PET.